on the question of how long it would take for something new to occur by an evolutionary means, we zeroed in on the very smallest level, which is a single gene producing a single protein molecule that folds up and does something useful inside a cell. And I zeroed in on this as a graduate student and decided I was going to do my postdoc on this question because I saw here's where you can actually go in the lab and measure the probabilities, and you could do the math and find out how long it would take for something like this to evolve. Whereas if you look at a whole organism, that's way beyond, we can't get the numbers. It's way too complicated. So I and a number of colleagues, Ann Gager, one of them, have spent years in the lab looking at how constrained the sequences are, the individual gene sequences, in order to make enzymes that work. And it turns out they're highly constrained. The amino acids have to be in a special order in order for the chains that are made inside the cells to fold up and to function. You can change these amino acids and still have a functional enzyme, but it turns out you can't change them very far. You certainly can't change them willy-nilly. There's a very tight constraint on what they need to be in order for these sequences to work. So the question we've asked is, suppose you've got genes that produce certain proteins that work, how hard would it be to evolve through just normal physical processes a new version of the gene that does some other function? And it turns out that if you need to make more than just a few, really two in most organisms, you're already beyond the probabilistic resources of evolution. You're already beyond the point where something could happen by accident that would make that transition. And we have studied a particular A to B transition where it appears that it's way more than two that are needed. We never did get it to work. Obviously, it could work. If you did hundreds of changes to A, you could turn it into B, and then it clearly would perform the function of B. But we were asking, can you get that sort of transition with just one or two? And you can't. And once you get beyond two, you get stuck. And that's even more true in higher life. So if you look at humans having supposedly evolved from the great apes, there you have much, much smaller populations, way fewer organisms than in bacteria, where you have billions and billions and billions, and you have much longer lifespans, and you have to come up with more sophisticated things. We differ from chimps in more profound ways than different bacteria differ from each other. So in all these respects, the problems that we find in the lab on single genes that work in bacteria only point to far greater problems when you look at higher and higher things like the origin of humanity.